I want you to look at your hand for a second. Just hold it up and look at the skin.
Okay.
It looks peaceful, right? Maybe a little dry, maybe a paper cut, but generally speaking, it looks like a quiet landscape.
It's the ultimate illusion.
It really is.
Yeah.
Because if we could switch our vision to a microscopic level, if we could zoom in about 1,000 times, that quiet landscape would look like, I don't know, the beaches of Normandy.
Yeah, that's not far off.
We are essentially talking fortresses
Yeah.
under a constant, relentless siege.
That is the absolute truth. I think we forget that we live in a biological war zone. I mean, every single breath you take, every time you scrape your knee against a desk, every bite of that sandwich you had for lunch, you are introducing potential invaders.
Constantly.
Bacteria, viruses, fungi, parasites, they are literally everywhere. They're on the microphone I'm speaking into, they're on your steering wheel, and they are all, trying to get in.
It's kind of terrifying when you frame it that way. It makes me want to go live in a bubble.
Well, the reality is, without the systems we're going to talk about today, you wouldn't last a week, even in a bubble.
Right.
We are constantly exposed to lethal agents, things like pneumonia, streptococcal infection, typhoid. These aren't rare occurrences in the grand scheme of nature. They are knocking on the door every second.
Every single second.
The only reason we aren't sick or, dead every single day is because of this hidden army that's just patrolling our blood vessels.
And that is exactly what this deep dive is all about. We were pulling apart the mechanics of human immunity, specifically looking at the resistance of the body to infection. We're going to be talking about leukocytes, better known as white blood cells, the massive monocyte-macrophage system, and the chaotic, destructive battlefield of inflammation.
This is one of my absolute favorite topics in physiology because it's so dynamic. It's not just anatomy, it's military strategy, it's logistics, it's chemical warfare.
It's all of it. So our mission today is, well, it's pretty ambitious. We aren't just going to list the cells. We're going to try to explain every single line of defense detailed in the source material.
From birth to death.
From the moment these cells are born in the bone marrow to how they learn to hunt, all the way to their final sacrifice in, and I apologize in advance for the visual, a pus-filled wound.
It's a messy business protecting the body, but someone has to do it.
Someone's got to do it. So let's start with the troops, the mobile units. The text refers to leukocytes as the mobile units of the body's protective system.
Right.
Now, help us understand the distinction here. I mean, a kidney cell stays in the kidney. A liver cell stays in the liver. What makes these guys so special?
Mobility, that's the defining characteristic. I mean, you're right. Most cells in your body have a fixed address. They are part of the architecture.
Right, they're the bricks.
They're the bricks. But leukocytes, they have a license to travel. They are formed partially in the bone marrow and partially in the lymph tissue, but once they're made, they hit the highway, the bloodstream, to get to wherever the trouble is. They are the rapid response team.
I love the statistics in this source material because they really put the scale into perspective. It says an adult human has about 7,000 white blood cells per microliter of blood.
Right. 7,000, which sounds like a lot.
It does, until you compare that to red blood cells. We have 5 million of those per microliter. So the white blood cells are really the elite few, aren't they?
They are.
They are vastly outnumbered by the civilians.
That's a great way to put it. They are the special forces. They're outnumbered by the red blood cells by a massive margin, something like 700 to 1. But their job isn't to carry oxygen. It's to seek and destroy. And because there are fewer of them, they have to be smarter, faster, and frankly, more aggressive.
So let's meet the lineup. The text lists 6 specific types of these warriors, and they all have very different roles. Walk us through the roster. Who are we working with?
Sure. So imagine a police lineup. First, you have the heavy hitters, the most numerous ones. Those are the neutrophils.
Neutrophils, okay.
They make up about 62% of all your white blood cells.
So the majority.
The infantry.
Exactly. The standard infantry, they are the grunts. They are the first ones into the fray, and as we'll see later, they're often the first to die.
Okay.
Then you have the eosinophils, about 2.3%. These are specialist units. We'll talk about their role with parasites later. Then the basophils, which are rare, only about 0.4%.
These are the ones involved in allergies, right? I've heard of them.
Yes, they are the chemical signalers, the ones that cause all the trouble with hay fever. Then you have the monocytes at 5.3%.
Okay.
These are the ones I find most fascinating. They're the transformers of the group. They change.
Ooh. And finally, the lymphocytes, which make up about 30%. These are the intelligence officers dealing with antibodies and long-term immunity, the memory of the system.
And the text mentions platelets here too, right? Just briefly.
Briefly, yes. Platelets are fragments of a larger cell called a megakaryocyte. There are tons of them, about 300,000 per microliter, but their main job is clotting, patching holes in the wall.
So they're more like the engineers and construction crew.
Exactly. Today we're really focused on the fighters, the immunity cells.
Okay, so we have our cast of characters. Let's talk about their origin story.
Yeah.
Because they don't just appear out of thin air. Where are these soldiers born?
It's a tale of two lineages. Imagine a family tree splitting right at the trunk. It all starts with the multipotential hematopoietic stem cell.
Okay, that's a mouthful.
It is. It's the great-grandfather of everything in your blood. But then it splits into two distinct paths.
And what's the first path?
First, you have the myelocytic lineage. This happens exclusively in the bone marrow.
In the marrow, okay.
This line begins with a cell called the myeloblast. This lineage is responsible for creating the granulocytes. That's your neutrophils, eosinophils, and basophils in the monocytes.
So the heavy hitters and the infantry all come from the marrow.
Correct. The marrow is the factory for the frontline troops. The second branch is the lymphocytic lineage. This begins with the lymphoblast. These cells are produced in the various lymph tissues, the lymph glands, the spleen, the thymus, the tonsils, and these little pockets in your gut called Peyer's patches.
So they're born all over the body in these strategic locations.
Exactly. They're more distributed.
Now, there was a detail in the text regarding storage and logistics that honestly blew my mind. I had to stop and think about the implications of it.
I think I know which one you mean. The warehouse concept.
The warehouse, exactly. The bone marrow isn't just a factory, it's a warehouse.
It is. This is a fascinating logistical feat. The body knows that an infection can strike instantly. If you get a cut, bacteria enter in milliseconds. You can't wait for the factory to spin up production from scratch.
No, it'd be too late.
That would take days, and by then you'd be dead. So the bone marrow stores about 3 times as many white blood cells as are currently circulating in your entire bloodstream.
3 times as many, just sitting there.
Just sitting there, waiting. Think about that mass. It's essentially a 6-day supply of soldiers, fully armed and armored, sitting in the marrow, ready to be deployed the second the alarm bells ring.
That is incredible forward planning, but it also seems expensive, biologically speaking. I mean, keeping a standing army that size, feeding them, maintaining them, just in case.
It is metabolically expensive. It takes energy to keep those cells alive. But the cost of not having them is extinction. It's the ultimate insurance policy.
Right.
Evolution has decided that the energy cost of maintaining that reserve is worth it to prevent sepsis.
That makes sense. Better to pay the utility bill than have the house burned down. But once they get deployed, I mean, the clock starts ticking, doesn't it? Let's talk about lifespans, because these aren't immortal cells. They aren't like neurons that stay with us for decades.
Far from it. Their lives are actually incredibly short, which adds to the tragedy and drama of it all. Let's look at the granulocytes, the neutrophils, for instance.
The infantry.
Yeah, the infantry. Once they leave the marrow and enter the blood, they only circulate for about 4 to 8 hours.
That's it. A single workday.
That's it. If they aren't called into tissue, they just circulate for a few hours. Then they move into the tissues where they might be needed, and they last maybe another 4 to 5 days.
And that's assuming peacetime.
Exactly. That's the best case scenario. In a severe infection, that lifespan drops to just hours. They rush to the battle, they fight, and they die in the process. They are essentially single-use weapons.
Wow.
They're designed to be expended.
That's a sobering thought. Millions of cells born just to die in a matter of hours.
What about the monocytes? You call them the transformers. Do they live longer?
The monocytes are a different story. They have a short transit time in the blood, maybe 10 to 20 hours. But, and this is the key, once they enter the tissues, they don't die. They swell up, they get huge, and they become tissue macrophages, the big eaters.
The big eaters. In this form, as macrophages, they can live for months.
Months?
Yeah, they act as sentries, stationing themselves in the tissues, just waiting for something to go wrong. So while the neutrophil is the disposable infantryman, the macrophage is the veteran soldier manning the outpost for the long winter.
And the lymphocytes, the intelligence officers.
They're the travelers.
Yeah.
They have constant recirculation. They go from the lymph to the blood to the tissue, back to the lymph. They can live for weeks or months, depending on the need. They are constantly patrolling the perimeter, checking IDs.
So we have this standing army in the marrow, we have sentries posted in the tissue, and we have patrols in the blood. But here's the problem I'm visualizing.
Okay.
The marrow is in my femur or my sternum. The infection is in my big toe. How do they actually get to the enemy? They don't have GPS, they don't have eyes.
This is where the physics and chemistry of the body get really, really cool. This is about movement and intelligence.
The text mentions 3 specific modes of movement. The first one is diapedesis, which sounds like a magic trick.
It basically is a magic trick. Imagine you are in a hallway. That's the blood vessel. The enemy is in the room next door, the tissue.
Okay.
But the door is locked. The only way out is through these tiny pores in the wall between the endothelial cells. But here's the catch. The pore is smaller than you are.
So you're stuck. How do you get out?
A normal cell would be a red blood cell, for instance, can't just leave whenever it wants, but a white blood cell can perform diapedesis. It pushes a tiny part of itself, a pseudopod, through the pore.
Like a little foot.
Like a little foot. Then it literally slides its internal contents, its cytoplasm, even its nucleus, through that tiny constriction until the whole cell pops out on the other side.
It's like an octopus squeezing through a jar opening.
That's a perfect analogy. It requires the cell to be incredibly flexible. The nucleus, which is usually a hard center, has to be lobulated. That's why neutrophils have those weird-shaped nuclei so it can squish through the gap. It's a momentary constriction to slide through the capillary wall.
Okay, so they've squeezed out of the blood and into the tissue. Now what? They have to move through the glorious mess of our body's tissues. It's not empty space. It's a jungle of collagen and fluid and cells.
That's where amoeboid motion comes in. They don't swim, they crawl.
They crawl.
Yeah. They project a little foot, a pseudopod, and it attaches to the surrounding tissue. Then they pull the rest of the cell body forward. Some of them can move at 40 micrometers per minute.
Which sounds incredibly slow to us.
But on a cellular scale, that's blistering speed. That's moving their own body length every single minute.
Wow.
Imagine if you could run your body length every minute through a dense forest. It's impressive.
But the big question remains, direction. How do they know which way to crawl? How do they find the bacteria in this jungle?
They have something better than eyes. They have chemotaxis. They can smell the battle.
The scent of battle. I like that.
Literally. When tissue is inflamed or when bacteria are present, they release chemical substances. Bacterial toxins, degenerative products of inflamed tissue, complement complexes, all sorts of things. Okay.
These chemicals drift out from the source, creating a concentration gradient.
So the smell is stronger, closer to the wound, and weaker further away.
Exactly right. And the white blood cells have receptors that can detect this difference in concentration across their own bodies. If the front of the cell smells more toxin than the back of the cell, it moves forward.
It just follows the scent.
It's magnetic. It can't resist it.
And what's the range on this? How far away can they smell it?
The text notes the effective range is up to 100 micrometers away from the source.
That doesn't sound like much. 100 micrometers.
It's huge in the microscopic world. Think about it. No tissue cell in your body is ever more than about 50 micrometers away from a capillary. We are incredibly vascular.
Oh, I see.
So if a signal goes out 100 micrometers, it is guaranteed to hit a blood vessel. It summons the hordes from the nearest road. There is nowhere for the bacteria to hide. Okay, so let's visualize this. The alarm rings. The cells smell the toxin. They stick to the wall, squeeze through the pores diapedesis, and crawl amoeboid motion toward the highest concentration of the smell. They've arrived. Now it's time for the violence.
Phagocytosis.
Eating.
Cellular eating. But it's not random. This is a crucial point that I want to emphasize. If these cells just ate everything they touched, they'd eat us.
They'd eat our own healthy tissue.
Right. They would destroy our own healthy tissue. So there is a very strict selection process.
Okay. The text lists 3 criteria for this menu selection.
Yeah.
How do they decide what's food and what's friend?
First, surface texture. They prefer rough surfaces. Natural, healthy body tissues are smooth. Invaders or dead tissues tend to be rough and irregular. It's a tactile check.
So if it's smooth, let it go. If it's rough, attack.
Roughly speaking, yes. It's one of the first clues. Second, protective coats. Most natural substances in our bodies have protective protein coats that repel phagocytes. It's like a do not eat label.
A biological friend signal.
Exactly. Invaders or dead tissues often lose these coats. They are naked to the enemy.
And the third one, this sounded a bit more complex, opsonization.
This is where the immune system cooperates. It's teamwork. We have antibodies molecules produced by those lymphocytes we mentioned. These antibodies attach to the invader. It's like putting a neon target on their back.
Highlighting the target.
Precisely. The antibody links the bacteria to the phagocyte, specifically utilizing the C3 product of the complement cascade. It creates a physical handle for the phagocyte to grab onto.
It's a tagging system. Eat this one.
Exactly. This one's approved for destruction.
Okay, target acquired. How do they actually eat it? It's not like they have mouths with teeth.
It's terrifyingly efficient. Let's look at a neutrophil. It attaches to the particle, then projects pseudopodia, those false feet around the particle. They embrace it.
Like a hug of death.
A total hug of death. They fuse on the other side, creating a chamber.
A trap.
A trap called a phagocytic vesicle, or phagosome. The bacteria is now inside the cell, floating in a bubble of its own doom.
And then the digestion begins, the stomach of the cell.
Exactly. Lysosomes and other granules inside the neutrophil come over and fuse with that bubble. They dump digestive enzymes into it. It's like pouring acid into a locked room.
The text makes a distinction here between neutrophils and macrophages regarding what they can digest. Why does that matter?
It matters for specific diseases. It's about specialization. Neutrophils mostly use proteolytic enzymes, good for digesting proteins.
Right.
But macrophages, they are the heavy-duty disposals. They have lipases.
Lipases digest fat.
Lipids, yes. And this is critical for things like tuberculosis. The tuberculosis bacteria has this thick lipid membrane, a waxy coat. A neutrophil can't get through it. It would choke on it.
It's too tough.
But a macrophage can digest that thick, waxy coat. That's why macrophages are essential for fighting TB.
Wait, digestion is one thing. That breaks down the body of the bacteria. But sometimes the bacteria is still alive in there. You need to kill it before you digest it.
Correct, you do. And this is the kill shot. This is one of the most, I think, metal things the human body does. It's chemistry at its most violent.
Explain this to us, because we usually think of our bodies as gentle, life-sustaining environments.
Not inside a phagosome. Not at all. The cell contains an organelle called a peroxisome. It generates oxidizing agents. We're talking superoxide, 2-$, hydrogen peroxide, H2-2-day.
Hydrogen peroxide, like what we put on cuts to bubble up and clean them.
Exactly, but inside the cell, in a concentrated dose, and it gets crazier. There's an enzyme called myeloperoxidase. It catalyzes a reaction between that hydrogen peroxide and chloride ions.
Chloride ions and salt.
Yes. And when you mix hydrogen peroxide and chloride with this enzyme, you form hypochlorite.
Hypochlorite. Wait a minute, isn't that bleach?
It is bleach.
Our cells make bleach inside themselves.
Your white blood cells essentially manufacture industrial-strength bleach inside that little bubble to kill the bacteria. It is extremely bactericidal.
That is profound. We have tiny bleach factories patrolling our blood vessels.
It shows you how dangerous these invaders are. The body doesn't play nice. It doesn't ask the bacteria to leave politely. It uses chemical warfare. It burns them with bleach and digests them with acid.
Let's step back and do a comparison, because we keep mentioning neutrophils and macrophages. Section 4 of our outline puts them head-to-head. Neutrophil versus macrophage. How do they stack up in a fight?
Okay, so think of the neutrophil as the infantryman. It matures in the blood, it's fast, it's aggressive, it's the first on the scene. But it has limits. It can eat maybe 3 to 20 bacteria.
And then it dies.
It exhausts its energy reserves. It fills up with waste products it can't get rid of. It's a kamikaze unit. Its death is actually what forms a lot of the pus we see later.
Okay, a short, violent life. And the macrophage?
The macrophage is a tank.
Right.
It starts as a monocyte, which is immature, but once it gets into the tissues and swells up, remember, it grows to like 5 times its size, up to 80 micrometers. It becomes a beast.
A beast, okay.
It can eat up to 100 bacteria.
100. So it's 5 times as effective as a neutrophil. Or more.
In terms of capacity, yes. And not just bacteria. It can eat entire red blood cells. It can eat malarial parasites. It engulfs things the neutrophil couldn't even dream of handling.
And does it die after eating, like the neutrophil?
Not necessarily. That's the other huge difference. It can extrude the waste products, spit out the bones, so to speak, and survive. It can keep fighting for months.
Wow.
It has a much more robust metabolic system. It's built to last.
Impressive. So we have the mobile units. But the text also talks about a static defense system. This is section 5, the reticuloendothelial system or the monocyte-macrophage system.
Right. If the white blood cells in the blood are the mobile army, the reticuloendothelial system represents the fortifications, the castles. These are macrophages that have stationed themselves in specific tissues to catch invaders before they can spread.
So it's a filter system.
It is. And it's positioned exactly where the enemy is most likely to attack. It's brilliant strategic placement.
Let's take the tour of these fortifications, starting with the outermost layer, the skin.
The skin. We have histiocytes. These are local macrophages living right there in the skin and subcutaneous tissue. If you get a cut, they are the first responders. They're literally sitting there waiting for the breach.
So they're already on site.
Already there. And they can divide in situ, meaning they can make more of themselves right there in the skin without waiting for the marrow.
Okay, moving inward. What if something gets past the skin?
It usually enters the lymph fluid. You have to remember particulate matter like dirt or clumps of bacteria can't get into the blood capillaries easily. The pores are too small, so it flows into the lymph vessels, and that leads to the lymph nodes.
The text describes these as having meshworks of sinuses. That sounds like a maze.
Imagine a hallway filled with spider webs, and in every web there's a spider. The spider is the macrophage. The lymph fluid has to flow through these sinuses, and the foreign particles get trapped in the mesh. The macrophages reach out and eat them.
So the lymph node acts as a scrubber, a filter.
Precisely. It prevents the infection from disseminating generally throughout the body. If you've ever had swollen glands in your neck when you have a sore throat,
Yeah, of course.
that's your lymph nodes doing their job. They are trapping the bacteria from your throat so it doesn't get to your heart or your brain.
Next stop. The lungs. Every breath we take brings in dust, pollen, bacteria. It's an open door.
It is. So we have the alveolar macrophages. They live in the alveolar walls, the air sacs. They digest almost everything. But, and here is a fascinating detail, what happens if you breathe in something indigestible, like silica dust or carbon particles from smoke?
Right. The macrophage can't digest rock.
No. It tries, but it fails. So instead of giving up, they do something amazing. They form a giant cell capsule around it.
What does that mean?
Several macrophages will merge together to form one massive cell, and they wall off the particle. They sequester it indefinitely.
So they put it in prison?
Life in prison. If you look at the lungs of someone who lived in a smoky city or a coal miner, you can see these little deposits. That's the macrophage just saying, we can't kill it, so we'll just hold it prisoner forever so it doesn't hurt the rest of the lung.
Wow. Okay, next organ. The liver. This one seemed incredibly high stakes to me when I was reading about it.
The liver is critical. You have to realize the food you eat isn't sterile. Your gut is full of bacteria. And the blood from your gut flows directly into the portal vein, which goes straight to the liver before it goes to the rest of the body.
So the liver is the gatekeeper between the sandwich I ate and my general circulation.
It's the bouncer at the club door.
Yes.
If the liver fails, bacteria from your gut enter your heart and brain. It's that simple. The liver sinusoids are lined with Kupffer cells. These are macrophages, and they are terrifyingly fast.
How fast?
They can phagocytize a bacterium in 0.01 seconds.
1/100th of a second. That's not possible.
It has to be that fast because the blood is rushing through the liver. If they were slow, the bacteria would sweep past them. They have to grab it from the moving stream instantly. The text says this filtration system is essentially 100% effective. Almost nothing gets past the liver into the general circulation.
That is humbling.
Yeah.
It basically means we are drinking a poison milkshake every day, and the liver cleans it up before we even notice.
And finally, if something does get into the general blood circulation, maybe from a cut that bypassed the lymph or a failure in the lung barrier, then you have the spleen and the bone marrow acting as the final filters.
The spleen is often thought of as a useless organ, but it's actually a blood scrubber, right?
It's vital. The spleen has a meshwork called the red pulp. Blood squeezes from capillaries into the cords of red pulp. It must pass through a meshwork to get to the venous sinuses.
So it's another physical trap.
Another one. Old abnormal red blood cells or debris get physically trapped in the mesh and eaten by macrophages. It's a final safety net for the bloodstream.
So it's a comprehensive system. Skin, lymph, lungs, liver, spleen. Every entry point is covered.
It's a defense in depth. If one line fails, the next one is there to catch whatever gets through.
But sometimes the invasion is too big. Sometimes the fortress walls are breached, and that's when chaos breaks out. This brings us to Section 6, inflammation. The battlefield.
Inflammation is often misunderstood. We think of it as a bad thing. Swelling, pain, redness. We take ibuprofen to stop it.
Right. It's a symptom to get rid of.
But it's actually a strategic response. It is the body preparing the ground for war.
What's the trigger? What kicks it all off?
Tissue injury. It could be bacteria, trauma, heat, chemicals. When cells are injured, they break open and release a whole cocktail of substances. Histamine, bradykinin, serotonin, prostaglandins, lymphokines. These are the chemical alarms.
And they cause the 5 characteristics of inflammation. Let's run through them, but explain why each one happens. It's not just random.
Not at all. Number 1, vasodilation. The blood vessels open up to bring more blood to the scene. That causes the redness and heat. You need supply lines open.
Makes sense. More troops, more supplies.
2, increased permeability. The capillaries get leaky. This allows the fluid and proteins to spill out into the tissue. 3, clotting of fluid. Fibrinogen leaks out and clots in the tissue spaces.
We'll come back to that one.
4, migration of granulocytes and monocytes. the army arrives. And 5, swelling of tissue cells.
I want to focus on number 3 for a second. The clotting. The text calls this the walling off effect.
Yeah.
Why would the body want to clot the fluid around the infection? Seems counterintuitive.
To quarantine it. The fibrinogen clots block the tissue spaces and the lymphatics. It essentially builds a wall around the bacteria. It says, you can have this cubic millimeter of tissue, but you cannot go any further.
It sacrifices the local tissue to save the whole body.
It's a containment strategy. And there was a really interesting comparison here between Staphylococcus and Streptococcus, 2 very common bugs.
Yeah, tell us about that.
Yes, the tale of 2 bacteria. Staphylococcus staph infections release lethal toxins very, very quickly.
Which sounds bad. Really bad.
Yeah. It is bad locally. But because it's so intense, the inflammation response kicks in instantly. The body panics and walls it off rapidly.
Aha, so the intensity is a good thing in a way.
It's a loud alarm. The result is you get a nasty contained abscess. It hurts, it's gross, but it usually doesn't spread.
Okay, and strep.
Streptococcus is sneakier. It doesn't cause intense local destruction initially. It's quieter, so the walling off process is slow. The body doesn't realize how bad it is yet.
It's a stealth attack.
It is. And the result is the bacteria can spread through the body before the walls go up. And that's why strep has a higher tendency to cause widespread death than staph, even though staph seems more violent locally.
That's the paradox of the walling off effect. Sometimes a big, loud reaction is better because it triggers a faster containment.
Exactly.
Now let's get into the timeline. Section 7. The 4 lines of defense. This part of the text reads like a military operation log. We have a massive infection, say, a crushed tissue infection. What happens hour by hour?
We can break it down chronologically. It's a beautifully staged response.
Okay, first line of defense, minute 0.
This is immediate. It's the tissue macrophages that were already there. They are the local beat cops. Within minutes of the injury, they enlarge, they break their attachments to the tissue, and they become mobile. They start eating the bacteria immediately.
But there aren't many of them, right? It's a small force.
No, maybe a few hundred in the immediate area. They can handle a skirmish, but they can't handle a full invasion. But they are life-saving in that first hour because they slow the enemy down while help is coming.
Second line of defense. The cavalry arrives.
This happens within the first hour. Neutrophil invasion. The inflammatory cytokines, like TNF and IL-1, released by the injured tissue, signal the blood. Neutrophils start sticking to the capillary walls. That's called margination.
Sticky walls, so they just start piling up on the inside of the blood vessel near the fight.
Yes, and then they squeeze through diapetesis and rush toward the chemotaxic signal. And suddenly you have an explosion of troops. Within 4 to 5 hours, the neutrophil count in the blood can jump from 4,000 to 25,000 per microliter.
That's the bone marrow dumping its reserves.
That's the 6-day supply we talked about getting unleashed. The floodgates open.
Okay, so the grunts are on the field. What's the third line of defense?
This is the second macrophage invasion. Monocytes enter the blood and then the tissue. But remember, monocytes are immature. They need time to swell and become macrophages. This takes 8 hours or more.
So they are late to the party.
They are late, but they are crucial for the long haul. It takes days for them to become dominant. But once they do, they are the heavy hitters for the cleanup. They eat the dead neutrophils, they eat the debris, they help rebuild the tissue, they're the cleanup crew, and the tanks rolled into one.
And finally, the fourth line of defense.
This is the factory reset, the bone marrow production. The activated macrophages in the inflamed tissue release growth factors, things like GCSF, GM-CSF, MCSF. These are hormones that travel to the bone marrow and scream, make more.
Because it's a feedback loop. The fighters on the ground call back to the factory for reinforcements.
A powerful feedback loop. It takes 3 to 4 days to really ramp up production, but once it does, the marrow can churn out cells for months to sustain the fight. If the infection lasts for weeks, this is what keeps you alive.
So to recap the timeline, 1, local macrophages, minutes, century.
Yeah.
2, neutrophil reserve dump, hours, the rapid response.
Yep.
3, monocyte maturation days, the heavy armor.
Right.
And 4, new marrow production, 3 to 4 days, the war machine. It's a perfectly staged escalation.
It's brilliant engineering. It ensures you use the fastest resources first and the most sustainable resources last.
But sadly, even with brilliant engineering, there is carnage. Section 8, the aftermath.
Pus.
Yeah.
We have to talk about it.
I know nobody wants to discuss pus while they're driving to work or cooking dinner.
Yeah.
We have to respect what it actually represents. What is it?
It's a battlefield graveyard. It is a mixture of necrotic tissue, dead neutrophils, dead macrophages, and tissue fluid.
It's the dead bodies of the war.
Literally. When you see pus, you are seeing the millions of neutrophil soldiers that gave their lives to kill the bacteria. It's a monument to their sacrifice.
Wow, I'll never look at it the same way again.
After the infection is suppressed, the dead cells and tissue gradually autolyze, they digest themselves, and the fluid is absorbed into the surrounding tissue. The evidence of the battle disappears.
Unless it's a really big battle, in which case it drains out.
Correct. Or it gets walled off into a cyst.
Okay, we've covered the main army, but we have some special agents we skipped over earlier. Section 9, eosinophils and basophils.
The special ops teams.
Let's talk about eosinophils first. They're only 2% of the blood. If they are weak phagocytes, meaning they're bad at eating, what is the point of them?
They have a specific nemesis. Parasites.
Like worms.
Schistosomiasis, trichinosis, hookworms, things that are too big to eat. You can't phagocytize a parasitic worm. It's thousands of times bigger than a cell.
Right. It would be like you trying to eat a whale in one bite.
It's impossible.
Yeah.
So what does the eosinophil do? If it can't eat it, how does it fight it?
I don't know.
How? It attaches to the surface of the worm and releases substances. It drops bombs,
Gun.
chemical bombs, hydrolytic enzymes, reactive oxygen, and a special polypeptide called major basic protein.
Which is what?
It's highly larvicidal. It dissolves the worm's skin. It's chemical warfare against giants. The eosinophils swarm the parasite and dissolve it from the outside in.
That is gruesome and amazing. The text also mentions they have a role in allergies.
Yes. They collect in tissues prone to allergies, like the lungs in asthmatics or the skin. Interestingly, it seems they might be there to help, to clean up. They detoxify inflammation-inducing substances released by mast cells. They try to dampen the allergic reaction to prevent excess damage.
Speaking of the mess, let's talk about the ones causing it, basophils and mast cells.
These are cousins. Basophils are in the blood. Mast cells are in the tissue right outside the capillaries. They are the alarm system.
The sentinels with the big red button.
Exactly. And what's their weapon?
Histamine.
Secretions. Heparin, which prevents coagulation, and the big one, histamine, along with bradykinin and serotonin.
And this is connected to antibodies again.
Specifically, the IgE antibody. This is the allergy antibody. It attaches to the mast cell membrane. When an antigen, say pollen or a bee sting venom, hits that antibody, the mast cell literally ruptures.
It explodes.
It degranulates. It dumps its load of histamine instantly. That causes the vascular dilation, the redness, the swelling, the classic allergic reaction.
So the runny nose, the swollen eyes, the closing throat. That's the mast cell doing its job a little too enthusiastically.
Precisely. It's reacting to pollen as if it were a deadly parasite. It's calling in the airstrike on a false alarm.
We're nearing the end of our deep dive, but we have to address Section 10, clinical abnormalities, when the system breaks. Because we've talked about how perfect the system is, but when it fails hard.
It is catastrophic. There is no middle ground.
First, leukopenia. Too few white blood cells.
This happens when the bone marrow stops producing. It can be caused by gamma rays from a nuclear blast, chemicals like benzene, or certain drugs like chloramphenicol.
What's the consequence? If
Well-
I stop making white blood cells today, what happens?
It's terrifyingly fast. The text says within 2 days after the marrow stops, ulcers appear in the mouth and colon.
Why the mouth? That's so specific.
Because, as we discussed, your mouth is full of bacteria right now, but your neutrophils are constantly fighting them back in the mucosal lining. Remove the army, and the resident bacteria invade immediately.
And then?
Respiratory infections. Without treatment, death occurs in less than a week. You die from overwhelming infection by bacteria that are normally benign. You don't need a superbug to kill you. Your own gut bacteria will do it if the army stands down.
That puts into perspective how hard those cells are working every day. We don't even notice them until they're gone.
Exactly. It's a thankless job.
The opposite problem is leukemia. Too many white blood cells.
Uncontrolled cancerous production. You can have lymphocytic leukemia, cancer of the lymph nodes, or myelogenous leukemia, cancer of the bone marrow.
But here's the paradox that always confused me. If you have too many white blood cells, shouldn't you be super immune? Shouldn't you be the healthiest person alive?
You would think it's a logical assumption, but these cancer cells are bizarre. The text uses that word. They are undifferentiated. They are half-baked. They don't know how to fight.
They're just bodies.
They're produced so fast that they never mature into functional soldiers. So you have millions of soldiers, but they don't have weapons, and they don't know who the enemy is. The patient has no protection.
And meanwhile, these useless cells are taking over the body.
They invade the spleen, the liver, the lymph nodes. They invade the bone, causing fractures because the marrow expands so much it cracks the bone from the inside. But the real killer, the text calls it metabolic starvation.
That phrase gave me chills. Metabolic starvation.
It should. These rapidly growing cancer cells are hungry. They are just dividing. They need amino acids, vitamins, energy to do that. And they steal it from the blood. They parasitize the body's resources.
So the cancer eats your food before your healthy cells can get it.
The body wastes away. Rapid deterioration. The cancer consumes the host from the inside out simply by being too hungry. It's a tragic biological greed.
It's a heavy note to end on, but I think it underscores the delicate balance of this system. It has to be aggressive enough to kill bacteria with bleach, but controlled enough not to starve the body.
It does. It's a tightrope walk every second of every day.
So, let's recap. We've gone from the genesis in the marrow, with its strategic 6-day supply, the warehouse. We've watched neutrophils squeeze through walls like octopuses and hunt via smell. We've seen macrophages act like tanks, eating hundreds of bacteria. We've toured the fortifications of the liver and lungs, seeing how they trap invaders in 0.01 seconds. And we've seen what happens when the balance tips into leukemia.
It's a complex, aggressive, intelligent system. It is the only reason we are sitting here today.
Here is my final thought for the listeners, something to chew on. We talked about how the body uses bleach hypochlorite inside our own cells to kill bacteria. We talked about how inflammation walls off parts of our own body, sacrificing tissue to save the whole. It seems like the core theme here is sacrifice.
It is a system of calculated sacrifice. The needs of the many outweigh the needs of the few cells.
But consider the metabolic starvation of leukemia. It's a chilling reminder that the cells meant to protect us. They are cut from the same cloth as the ones that can destroy us. The difference between a protector and a parasite is often just a matter of regulation. A cell that forgets its place consumes the world.
That is a profound thought. We survive only because of strict biological discipline. When discipline fails, the fortress falls.
And on that note, we're going to wrap up this deep dive. Thank you for listening.
Keep learning.